Neurons are the fundamental units of the nervous system, specialized to transmit information rapidly and efficiently throughout the body. The communication between neurons relies on a combination of electrical and chemical signals, with action potentials being the core mechanism of rapid electrical communication. An action potential is a brief, fast, and self-propagating change in the electrical charge across a neuron's membrane. This electrical impulse allows neurons to send messages from one part of the body to another, almost instantly, making movement, sensation, and reflexes possible. The process of generating an action potential begins when a neuron receives a stimulus strong enough to exceed a threshold. This could be anything from a touch, a sound, a change in temperature, or a chemical signal from another neuron. When the threshold is reached, depolarization occurs. During depolarization, voltage-gated sodium channels in the neuron's membrane open, allowing sodium ions to rush into the cell. This influx of positively charged ions changes the electrical potential inside the neuron, creating an upward spike in membrane voltage. This spike is what we recognize as the action potential. As the action potential moves along the axon, the neuron must return to its resting state to be ready for the next signal. This is achieved through repolarization, which occurs when potassium channels open and potassium ions flow out of the neuron. The outflow of positive ions restores the negative charge inside the cell, returning the neuron to its resting potential. In some neurons, a brief hyperpolarization phase occurs immediately after repolarization, where the membrane potential temporarily becomes slightly more negative than the resting state. This ensures that action potentials travel in one direction along the axon and prevents backward propagation of the signal. Many neurons are covered with myelin sheaths, which are insulating layers produced by specialized glial cells, called Schwann cells in the peripheral nervous system, and oligodendrocytes in the central nervous system. Myelin acts like electrical insulation around the axon, preventing the loss of electrical charge as the action potential travels. In addition, the action potential does not move continuously along a myelinated axon. Instead, it jumps between gaps in the myelin called the nodes of Ranvier. This jumping process is known as saltatory conduction, and it significantly increases the speed of signal transmission, allowing action potentials to travel at remarkable speeds, sometimes over 100 meters per second in large neurons. At the end of the axon, the action potential reaches the axon terminals, where it triggers the release of neurotransmitters, the chemical messengers of the nervous system. These neurotransmitters cross the synaptic cleft, the small gap between neurons, and bind to receptors on the next neuron or an effector cell, such as a muscle or gland. Conversion of an electrical signal into a chemical signal and then back into an electrical signal in the next neuron ensures that communication across the nervous system is fast, precise, and coordinated. Action potentials are not just important for reflexes or voluntary movement. They are fundamental to every function of the nervous system, including sensory perception, motor control, cognition, emotion, and learning. For example, when you touch a hot surface, action potentials travel from your skin receptors through sensory neurons to the spinal cord, initiating a reflex response even before the brain processes the information. At the same time, other action potentials carry the same information to the brain, allowing conscious perception of pain and memory formation. Similarly, thinking, decision-making, and voluntary movement all rely on the generation, propagation, and integration of countless action potentials across neural networks. The remarkable efficiency of action potentials is due to the combination of depolarization, repolarization, and myelin-mediated saltatory conduction. These processes ensure that signals can travel long distances within the body, rapidly and reliably, maintaining communication between the brain, spinal cord, and peripheral organs. Without action potentials, the body would be unable to coordinate muscle contractions, perceive sensory stimuli, respond to danger, or carry out even the simplest voluntary or involuntary actions. In addition, the nervous system's ability to transmit signals via action potentials is adaptable. Neurons can adjust their excitability based on prior activity, a process that contributes to learning and memory. Synaptic plasticity, the strengthening or weakening of synaptic connections, depends on the precise timing and frequency of action potentials. In this way, action potentials are not merely electrical impulses. They are the foundation for the complex processes that make thought, perception, and behavior possible. In summary, neurons transmit information through action potentials. Rapid electrical impulses generated by depolarization and repolarization of the neuron's membrane, myelin sheaths, and saltatory conduction enable these signals to travel quickly along axons. At the axon terminals, action potentials trigger neurotransmitter release, passing messages to the next neuron or effector cell. This process allows the nervous system to coordinate movement, reflexes, sensation, learning, and cognition almost instantaneously. SLOCs, the intricate design of neurons and the efficiency of action potentials, showcase the remarkable capabilities of the human nervous system, keeping the body connected, responsive, and alive every moment of our lives.